Today, it's a great honor to have with us Dr. Peng. Thanks also to the connection that Gabriele Codotto made with him at the Congress of the European Association for the Study of Liver some years ago when Gabriele was our student. Dr. Peng is currently the group leader at the Princess Maxima Center for Pediatric Oncology in Utrecht, Netherlands. He leads a program dedicated to understanding childhood liver cancer through omics technologies and hepatoblastoma organoid models. But I also want to mention that we first discovered his work in the literature when he was a postdoctoral fellow at Stanford University in the USA. Because Dr. Peng was one of the pioneers in developing hepatic organoid models using inflammatory cytokines-based approaches, in particular TNF-alpha. And during Gabriele's thesis here in FIF, we also developed this model in our laboratories in FIF, following Dr. Peng protocol. So I don't want to take up more time. I will hand the floor to Dr. Peng, and thank you for your participation.
Yeah, thanks. Let me share my screen.
Yes, you can share your screen.
Okay. Good. Well, thanks for inviting me, and thanks for the kind introduction. It's my pleasure here to speak to you all today. So I'll be mostly speaking about how we use organoid to advance our understanding on pediatric cancer research, well, especially in the hepatoblastoma, which is a childhood liver cancer. And so before I start, let me just quickly introduce the center to the audience here. The Princess Maxima Center is currently the only pediatric cancer in the Netherlands. Since a few years ago, the treatment of all children with cancer is now centralized in one center. The idea is that this would better, let's say, improve the care and treatment of these young patients. And we currently have about 1,000 people working in this place and treat about 600 patients every year. So this is also the largest pediatric cancer center in Europe. And Hans Klevers here was the first scientific director for our center. And right now we have about 40 different research groups working on different tumor types and technologies. And in general, pediatric cancer it's rare. You can see here the most common ones are leukemia, followed by brain tumors. So solid tumors like hepatoblastoma or other liver cancer is super rare. And you can also see this from this plot here of cancer incidence rate across different age group. So in the adult, this is typically HCC, but in young patients, this is predominantly hepatoblastoma. So HCC also occurs in young patients, but this is extremely rare. And here you see a spike in the cancer incidence rate right after birth, and then it plateaus down again. So because the cancer is normally, let's say, diagnosed in very young patients, this is considered a developmental disease, and hepatoblastoma has a very unique mutational landscape. Almost all tumors have mutation in the beta-catenin genes, resulting in stable proteins and accumulation of beta-catenin, which leads to high Wnt signaling. So Wnt signaling is the main key driver in hepatoblastoma. Other than that, it has very low mutational burdens. You can also see that from this publication several years ago that looks at genomics alteration across childhood cancers. Hepatoblastoma is here at the bottom of the list as one of the lowest mutational burden. However, the disease itself is considered very heterogeneous because of the diversity in histology. And here in the brown tumors, you see that the tumors appear more undifferentiated, and the cells are smaller, they have larger nuclei, whereas in the fetal ones, they are more differentiated, more hepatic-like, and the well-differentiated one is the least aggressive ones, right? Whereas the browner ones are more aggressive, they tend to be more proliferative. And the different histology subtypes actually resemble the different stages of liver development. And it's common to find these different subtypes, different entities within one tumors. And you can imagine this complicates diagnosis and disease treatment. So my lab has a long-standing interest in Wnt signaling as well as in the liver. So I started my PhD by studying Wnt signaling regulator such as LGR5, OSPONDing, and E2 ligase ZN-RF3 using X-ray crystallography. And then during my postdoc, I moved to Stanford to Roux Nussler lab and to study Wnt signaling in stem cell and regeneration. So there we developed the hepatic organoid models that was very useful also in studying liver regeneration. And then during that time also, we contributed to single cell analysis for liver tissues for the mouse cell atlas projects. So currently our research is mostly focused on understanding tumor origin and how they develop in the liver and what leads to this tumor heterogeneity. This is very important because the tumor heterogeneity affects clinical outcomes such as survival, relapse, and also response to chemotherapy. And here, before we talk about tumor biology, I just want to give a very brief overview of Wnt signaling in the liver. This is a liver lobule, and the hepatocytes around the central veins are very high in Wnt signaling. That's because the endothelial cells around the veins here, they secrete Wnt and OSPONDing. And as a consequence, the hepatocytes around here, they express metabolic markers that are driven by Wnt, such as glutamine synthetase. And the glutamine synthetase here, or GS, is shown in this figure. They are around the central veins. And the expression is, of course, niche dependent. It depends on the secretion of Wnt and OSPONDing around the veins, and is not expressed as outside of the central veins region. But in the tumor, because of the mutations, then it's broadly expressed, and this is not a niche-dependent process. And here, GS appear to be a very good markers, very specific markers for Wnt signaling in the liver. And we also often use it for tumor diagnosis. And here to look at tumor heterogeneity, then we use single cell analysis. And here we are able to distinguish tumor cells from different patients and group them into two main baskets. Here we call them fetal cells and also the embryonal cells. And if you compare them, the gene expression pattern, you will see that the fetal ones are enriched in liver-related functions, such as drug metabolism, bile acid metabolism, fatty acid, whereas embryonal ones are much more enriched in Wnt signaling and other, pathway that is more related to cancer development, such as TGF-beta signaling, TNF-alpha signaling, and they also display these EMT features. So overall, the embryonal ones are more progenitor-like. They appear to be more undifferentiated, whereas the fetal ones are more differentiated, more hepatic-like. And here we can also look at the gene expression pattern across the different subtypes. And here, Wnt signaling targets such as exon 2, NOTAM and NACA1 are highly enriched in the embryonal subtypes and also expressed to some extent in the fetal cells. They're usually very low and very hard to detect in the normal liver. And here, Wnt metabolic enzymes such as glutamine synthetase, GS, and OAT are high in both subtypes. And but other than that, hepatic markers like albumin or CYP2E1 are low in the embryonal ones. They are expressed in the fetal cells. In a way, they resemble mature hepatocytes to some extent. But we notice that compared to normal hepatocytes, the fetal tumor cells they express fetal markers such as glipin-3 and SPIN1. So these are also very good markers for differentiating tumor cells from normal hepatocytes. So by and large, expression pattern is consistent with Wnt high in embryonal cells, Wnt low in fetal cells, and hepatic markers in fetal cells. And here we look at transcriptional factor activities by performing this so-called gene regulatory network analysis. And here this is the transcriptional factors activity score. And we can also do a UMAP clustering, and you can see that the two subtypes have very different transcriptional signatures. So the TCF-left, which are the core Wnt signaling regulators, are very high in the embryonal subtypes, whereas the fetal subtypes are enriched with HF4 alpha and the FXR regulators. So HF4 alpha is normally considered a master regulator in liver differentiation. It's required for the progenitor cells to become hepatocytes, and it's also expressed in adult liver to maintain liver functions, whereas FXR regulates the glucose, bile acid, and lipid metabolism. And here, more recently, we did single cell and ATAC-seq to look at the epigenetic landscape. And here we were also able to detect, let's say, the embryonal classes and the fetal classes. And if you look at the chromatin accessibility profiles, I mean, the embryonal ones are enriched with these TCF-left motifs, whereas the fetal ones are enriched with HF4 alpha related motifs. So the two subtypes have very different epigenetic landscape. And now we're really curious if we can use then these two markers to visualize the different tumor entities within tumor tissues. And then we did that by using immunostaining. We stain a small cohort of patients, about 20 tumor samples, and this is a piece of a normal liver. And around the central veins, the Wnt is high, but we normally do not see left one positive staining in these cells. And all hepatocytes are, of course, positive for HF4 alpha. And here with the tumor tissues, then you see a very distinct subset of cells that are positive for left one. So these are the embryonal cells in orange and the fetal cells here in blue. And we never saw an overlapping between the two markers. So these two markers are really good in distinguishing the tumor subtypes. So this is for the first time that we can visualize the tumor entities by immunostaining of just using two markers. And the next question is, we are curious, where are the beta-catenin localized in these tumor tissues? In normal tissues, they are usually membrane restricted and also found in the cytoplasmic regions. But in tumors, because of the accumulation, lack of ubiquitination, they accumulate in the cytoplasm and then translocate to the nucleus to activate Wnt signaling. But staining for beta-catenin is usually not straightforward because they usually look very heterogeneous, and this is also what we saw. Here you can see that it's a very heterogeneous staining, not very clean, but with left one staining, we can see that the embryonal cells have higher beta-catenin intensity than the surrounding fetal regions. Right, in the fetal regions, it's much more diffuse, weaker, and in the bottom panel here, the more differentiated tumor tissues, we see a more membrane staining of beta-catenin. So we did some quantitative analysis as well, and by and large, the beta-catenin localization correlates with tumor differentiation. But the staining on its own, it's difficult to distinguish tumor subtypes. So we have to rely on HF4 alpha and HF left one end to distinguish the tumor entities. So just to sum up the key findings from these omics studies, here we have a Wnt high embryonal tumors with left one positive cells. They are more progenitor-like. They also display EMT features. And the fetal tumors are Wnt low, HF4 alpha positive. They're more differentiated, more hepatic-like, and there appear to be Wnt signaling dampening in fetal tumors, even though both tumor subtypes have the same mutational background. And this is likely happens through epigenetic regulations. And these findings, this observation help us to understand the tumor biology, but it's still very difficult to translate this knowledge into effective clinical application. And here we really need a good experimental models to study tumor development and to find new therapies, right? So here I wanted to switch gear to how we use organoid to study tumor biology and also for translational research. So when we started a few years ago, there was no good in vitro model for hepatoblastoma because they are rare and tumor material is very limited. So there are tumor cells like that such as HepG2 cells. This is actually a very good model for hepatoblastoma, not HCC. But based on what we know now, the HepG2 cells are more fetal-like and not embryonal-like. So we set out then to generate organoid models from clinical samples. Here we collected needle biopsy samples from patients at diagnosis. And then after chemotherapy, the tumors are usually resected. So we also collect tumor materials from patients. And if there's any metastasis in the lung, we also try to collect these tissues. And this is a piece of needle biopsy tissues. And we dissociate them into single cells or smaller pieces, and then we plate them in matrix shell for organoid culture. And so far, we have been quite successful in growing organoids from patient samples. It's much easier to grow, let's say, organoids from biopsy samples because the viability is much higher. And it's usually much more difficult to grow organoids from resected tissues because of necrosis, tissue necrosis. But we have been quite lucky that we were able to get organoids from 20 different tissue specimens. And these are, culture with, the standard culture media, with an osmotic, EGF and TGF beta inhibitor. The cytokines are not needed here. And here, based on the morphology, it's already possible to distinguish the tumor subtypes. The embryonal cells are more spheroid-like. They appear quite undifferentiated. The cells are very small, and they have very dense nucleus. And the fetal ones are more epithelial-like. They are more hepatic-like. They look like the normal hepatic organoids that we made using mouse cells. And they are also large. The cells are also somewhat larger. And usually we get one tumor subtype from one, let's say, tumor specimens. But occasionally also we get, let's say, the mixture of these different subtypes within the culture. So now to better understand the heterogeneity, we use single cell analysis. Again, here you can see that the clustering is mostly based on as patient-specific features. And this is a very common issue when working with human samples. There's so much heterogeneity between patients that the clustering that we see is mostly driven by these patient-specific features. So we try different ways, but what works best in the end is really just the PCA analysis and this the scenic analysis. Here we see different the clustering based on tumor differentiation state. For instance, here in embryonal clusters, we see Wnt high, hepatic low. And the fetal tumors, we see two clusters, one that has very high hepatic features and one that has intermediate hepatic features. And the pattern here is consistent with this, with the scenic UMAC clustering. So we can also examine an expression marker. Right here, the albumin, serpin A1, and apolipoproteins are high in the fetal cells. They also express GS and fetal marker such as glypican-3. Whereas in the fetal ones, you can see left one, DKK notum. The pattern is quite consistent with what we see in tumor tissues. There's a correlation with Wnt and stemness of the cells. So here we did single cell and ATAC sequencing analysis again. And the pattern is quite consistent with what we see in tumor tissues. TCF left in embryonal cells, HNF4 alpha in hepatic nuclear factors. And in the intermediate group, we also see this expression of more, let's say, inflammatory related pathway. And we also did immunostaining to confirm the expression of LEF1 HNF4 alpha. Here we often see organoids that are either positive for LEF1 or HNF4 alpha. We never see, let's say, an organoid which has both LEF1 positive cell or HNF4 alpha positive cells. And the organoid seems to maintain the phenotype over time. So meaning that we never saw LEF1 positive organoids becoming HNF4 alpha positive. And we also did not see HNF4 alpha positive cell becoming LEF1 positive. So it appeared to be quite epigenetically stable in culture, even after many, many months in culture. So this makes them a very good model for us, at least, to, let's say, to separate the tumor entities for studying, let's say, tumor biology and also for finding new drugs. Now, so we use these organoid models for finding new therapies. Here we do drug screening. We also use them for metabolomic studies, for proteomic studies, And we also collaborate with others for co-culture with engineered T cells. So this has been published last year by collaborators. And for this talk, I will focus on our findings using drug screening. And so here we use the organoids and expose them to about 200 different drugs. And the drugs here are mostly, many of them are FDA approved, and some of them are also in clinical studies. So this makes translation more straightforward for us. And we typically expose the cells for 5 days in culture, and then at the end of, expose the cells to the drugs for 5 days in culture, and then at the end of 5 days, then we measure the viability. And then the response is shown here in this heat map. So red means very sensitive, and blue means resistant to the treatment. And here in this region, you see that all organoids are sensitive to chemotherapy agents. There are also some compounds that are very effective for the different tumor subtypes. We also see that there are some subtype-specific drug response. So some membranous cells are specific to respond to some drugs, whereas fetal cells respond to a different group of small molecule inhibitors. And one of the key findings that we made from drug screening is that there is this group of molecules or compounds that they're very potent in our screen. This targets the histone deacetylase. So each tag is a family of epigenetic regulators. So they regulate chromatin remodeling and they regulate gene expression patterns. So, and now we can look at this compound separately. The romidazine here targets HDAC1 and 2, and femipinostat target 1 and 2, 3 and 10, and panobinostat is an pan-HDAC inhibitors. So the romidazine, which target 1 and 2, are very potent in our screen. We also find other promising molecules like PLK1 inhibitors and proteasome inhibitors. So these compounds have been used in clinical studies for other tumor types. Now we have these very nice findings, and to help us to understand which one is more selective for hepatoblastoma, so we compared the drug response to other pediatric cancer type. So here we compared the drug response to the screening data done by other groups here at the Princess Maxima Center. For example, the Wilm tumors, neuroblastoma, and also Ewing sarcomas developed by the Clayvers group. Here from this plot, based on the Z-score and the AUC value, which correspond also to the IC value, we see that romidazine, which target HDAC1 and 2, are highly selective for hepatoblastoma. So panobinostat is here, it's broad range, so therefore it's less sensitive for hepatoblastoma. And based on older studies, we also know that HDAC1 and 2 are upregulated in hepatoblastoma, and cell lines like HepG2, but are lower in the normal liver. And here we have access to bulk sequencing data for the original tumors that these are performed in a diagnostic setting. And here we also see that HDAC1 and 2 are overexpressed in a tumor biopsy, and this is higher than the other liver cancer, other tumor types in the liver, like adenoma or HCCs in young patients. Because with the organoid, we also check for HDAC expression by qPCR. Here you can see that both HDAC1 and 2 are quite highly expressed. This is by comparison to GAPTH, which itself is super high, so that's why it appears lower here. And
can you ping the windshield wipers down?
Sorry. Yeah, so here we see that the embryonal expression is has higher HDAC1 and 2 expression than the fetal ones.
So what would be good is if you go down to the sidewalk.
And
if you go down to the sidewalk so I can go out and you can tell me when it's safe, okay? Katie?
Yeah. I'll continue. Yeah.
No. Please, so, Julie, close your mic. I think Please, Julie, close your mic.
All right.
Sorry, Dr. Peng, you can go on. Sorry.
Yeah. So the next interesting observation that we made here is that when we compare the drug response between the two tumor subtypes, we see that a group of compounds that target EGFR-related family members are very potent against the fetal tumors, and compounds that target FGFR are more potent for the embryonal tumors. And here, this is a representative drug response curve. So afatinib targets mainly EGFR, and they are most sensitive for the embryonal, for the fetal tumor cells. And then the FGFR inhibitors here are more sensitive for the embryonal cells. And here we can use the organoid to ask the question whether the tumor cells are dependent on these growth factors, right? And here we drew EGF from the culture medium for 2 weeks, and you can see that the growth reduced by almost 50% after 2 weeks. And on the other hand, the brown tumor cells are not dependent on EGF in the culture medium. And then we went on to examine the expression pattern of EGFR in tumor tissues. Here we see a very nice overlap between EGFR and HNF4 alpha. In the middle region here, this is LEF1 positive cells. They're not positive for EGFR. And next, we also examine the FGFR1 expression pattern, FGFR expression pattern. So we also did that by staining, but we still do not have a good antibody for FGFR1. But from the single cell analysis, we see they are high in embryonal tumor cells. And the tumor also secrete different FGF ligands. And we see the same pattern in the organoids. Here by qPCR, FGFR1 is high in embryonal subtypes, and they also make multiple FGF ligands. So we know that the cells are not dependent on FGF, exogenous FGF in the culture media. So meaning that we did not have to add any FGF growth factors. So this is likely an autocrine signaling in the embryonal tumors. And so this is just to sum up our findings. The Wnt signaling itself is not sufficient to drive tumor progression. They also need growth factors and receptor tyrosine kinase. And in the liver, the FGF appear to be secreted by the tumor cells themselves, whereas EGF is normally secreted in the duodenum in the intestine, and they circulate to the liver directly via the portal veins. So EGF is also important for normal liver functions. And we think that the postnatal liver is still actively growing. They are rich in growth factors, and this may have helped the tumors to develop. And these findings also, generate insights into therapeutics, because there are already clinically approved, EGFR antibodies in the market. And we will be testing this using our organoid models. Now, this is the last few slides. I just want to mention how we use our findings in the lab for clinical application. Here, we started a collaboration with Ronald Krieger, who is a pathologist at the Princess Maxima Center. Now we're incorporating HNF4 alpha and LEF1 in our diagnostic pipelines here to help us to better classify tumors. And we are also looking forward to collaborate with the international community for large-scale study to validate these two markers for tumor classification. So this is currently one of the few tumors that do not have good biomarkers for classification. And we are quite excited about this, these findings. And the next thing that got us quite excited is using these drugs that we found in a clinical study. So the Maxima is very active in this International Childhood Liver Study group, also called the CellPath. And since 2002, we have been, sharing our data with the community. So we have regular meetings with other groups, other European groups, and our data, and as well as data from other groups, show that the panovidostat, or in general HDAC inhibitor, is very good against hepatoblastoma. So now we have all this data was published recently last year, in different journals, and we have data from organoids and also mice study demonstrating in vivo efficacy. And we're starting a clinical study to evaluate the efficacy of panovidostat in patients with high-risk disease, those who present metastasis in the lung at diagnosis. And the reason to go with panovidostat and not other HDAC inhibitors because this is approved by the EMA. And the study is led by Dr. Joseph Zeros, who is a pediatric oncologist at the Princess Maxima Center. And the studies will likely start next year. So we'll have the opportunity to collect patient samples and also to look at more in-depth into the HDAC mechanism in these patient samples. And with that, I would like to thank the member of the labs who did the study and also the collaborator, especially the clinicians who has been very helpful in pointing us to the right directions. And we are very excited that what we do in the lab can be translated in clinics within a very short time frame. So this is very exciting for us. And with that, I would like I'm happy to take any questions.
Thank you very much, Dr. Peng, for this seminar that was really very informative and also very clear.